Okay, Psych class, welcome to video number 2 for Unit 2C on Sensation and Perception. In today's video, we are going to discuss vision. How does your eyesight work? And obviously this would be, in the beginning, we'll talk about sensation components, like how the information actually gets from your environment into your brain, and then we'll have fun with the interpretation of that a little bit later. But let's look at the screen here is a kind of a review, right, from a science class, that what we see as light is only a tiny slice of the wide spectrum of electromagnetic energy. So, visible light to humans is just a very small part of all light, but that would be what we can sense in our environment. So important words that you need to know concerning light energy, right, from the environment into the brain. Let's talk about wavelength. And that is determining the hue or color that we experience. Okay, so short wavelength, right, is going to give you different colors than long wavelengths. And you can see that in part A in the chart below. Now, the intensity or the amplitude is going to give us bright colors versus soft colors, right? And so the wavelength will give you the color, and the amplitude or the height will determine the intensity of that color. Is the same is going to apply when we talk about sound, as you can see in parentheses there, but that's a video for another day. Okay. Other organisms can see different portions of the light spectrum. So like the flowers on the left to B, right? They can't see red, but they can see ultraviolet. So that's what those same flowers would look like to a bee, which is kind of cool. Okay, so light is coming into our eye. That is the portion of our body that receives visible sensations from our environment. And so when we talk about the eye, an important part that I do want you to know, and again, take anatomy if you want to learn all the parts of the eye, but it is important for you to know, again, these blue words that are floating across the screen. The retina is the inner surface of the eye. Okay, now we'll talk about these photoreceptors in another slide, right? But they're the neurons that begin the processing of visual stimuli. And so light is inverted by the lens and is interpreted by the brain as an upright vision. I have some examples of things like to show you in class to demonstrate what that process is like. But as long as you know the retina, you are good. Again, don't feel obligated to copy this down, but it's just kind of to show you what's happening, right? The picture, the image of the candle, right, enters the eye through the light enters the hole known as the pupil. Right, the iris, that colored part of your eye, is the muscle that actually helps open or close your pupil. Right, the protective outer surface is called the cornea. That lens is what inverts the image onto the retina, or that inner surface. The fovea is what we call the central focus point. We'll talk about blind spots here in a second. And then all that information is sent through these axons of these different neurons traveling to the brain, and we call that bundle of axons the optic nerve. Okay, so just a little overview there for you. There are no receptor cells where the optic nerve leaves the eye. I want you to know retina, and I also want you to know blind spot. And so the image there at the bottom can test your blind spot. And so what you need to do is, align your nose, to the center of this picture. And if you cover your left eye, and don't move your head, but use your right eye to stare at the plus sign, and then slowly, move closer and closer to your computer screen. There will come a point, I'm really close now, where that black dot disappears and just becomes white. Okay, that's because I've lined that, I stare at the cross, and I've lined my right eye up in a way that that represents my blind spot. If you're having a hard time with that, I do have some printouts that we'll use in class. Sometimes it is more helpful to see it on a piece of paper or cardstock than it is on the computer screen. So everyone has blind spots. Don't be alarmed. And if you can't get this picture to work, don't be alarmed. We'll play more in class. Okay. Inside the retina, I do want you to know the two different types of cells, rods and cones. Okay, rods are what are the cells that detect blacks, whites, and grays, and they're necessary for our peripheral vision and, like seeing at nighttime, we rely on our rods. That's why, like in a dark room, colors aren't necessarily as vivid as they are when in a well-lit space. The cones are the clusters near the center of the retina, and they help us see fine detail and allow us to see in color. So those of you that maybe experience color blindness, it's a result of a deficiency concerning some of the cones in your eyes. So this is easy to remember. Color cones, they both start with C. And we call them rods and cones, as you can see from the picture, just based on their shape, the shape of those neurons. Okay, light energy triggers chemical changes in the rods and cones, which activate bipolar cells, and the bipolar cells will activate the ganglion cells, which take it via the optic nerve. So the optic nerve are all the axons of these ganglion cells that transmit the neural impulse from the eye to the brain. Don't worry about the details of that process. I actually cut that slide out. No rods, no cones, and you're awesome for the intro to psych portion. Again, anatomy would cover this in more detail. Okay, related to those rods and cones, we can talk about what's known as an after image. So this is something kind of fun to play with. If you see that tiny red dot in the center of this flag and you stare at this for a number of seconds, press pause if you need to on your video, and then move towards the white part of the slide, you'll see this kind of floating after image of the American flag in the proper red, white, and blue colors. Okay, that's just a result of how these rods and cones work together in pairs and opposites, right? And so the opposite of yellow being the royal blue, right, and the opposite of the black being the white. You tire out a specific element of those rods and cones, and you get that what's left, right? The opposite or the after image. Okay, concerning looking at the various features, we're done with the eye. The information has traveled via the optic nerve to the brain, and we have that visual cortex located in the, do you remember? Occipital lobe. Hopefully you do remember that, right? And within that visual cortex contains these feature detection cells. So we have certain cells that are in charge of interpreting edges, lines, angles, or information, like passing it on to more complex processing in other parts of the brain. And brain scans can show us this, like based on what feature detection cells have been activated and where they're sending the information. We can look at a brain scan and know whether a person is looking at something like a shoe or a chair or a face because of the various feature detectors that become stimulated when that information is sent to the visual cortex. So that's kind of cool. The brain is going to use that parallel processing that we talked about in Unit 1 to assign different teams of cells to simultaneously process the many aspects of the scene. And so, like, for example, looking at a bird flying across, the sky, we've got feature detectors that are looking at the color of that bird, that are looking at the depth, at the motion, and at the shape or the form. Now, you don't really realize the separate cells doing this because parallel processing means that they're all working in tandem or they're all working together. Now, there have been cases, as there's one mentioned in your book, and we'll talk about her in class, Mrs. M, who was unable to perceive motion because a stroke damaged those certain feature detector cells, and that just meant that objects jumped from place to place. And so a very interesting example that we'll explore more in class, that like we don't realize how much information is being simultaneously processed by that very amazing brain of ours. Okay, so that's the basics on how the eye works and how vision is processed and sent to the brain, right? This is kind of this, in looking at the tiger in the center of our graph, you start at the bottom there. There's the scene. Here's what's going on in the retina, right? Our rods and cones are sending the information to the bipolar cells, which send it to the ganglion cells, which therefore sends it to the brain, and those special feature detector cells are going to respond to specific elements: the color, the shading of the tiger, the position of the tiger, the lines, the angles. Parallel processing means that we don't recognize that all this stuff is happening because it's all kind of combined together into one succinct image of the tiger. And then that leads to recognition, right? Our brain interprets that, sends that information out to the part of the cerebral cortex that stores my memory of tigers and allows me to recognize this as, hey, a picture of a tiger. So it's really cool that this very complex thing happens rather seamlessly to us, our brains. And so we'll talk about interpreting what we see or perception of what we see when we come to class next. So bring your notes with you, and along with your notes, any questions you may have concerning vision. Thanks for listening.